Tunneling nanotubes are thin nanoscale connections that cells in culture form to communicate with each other. And our lab has shown that these tubes, which we call TNT, allow the spreading of pathogenic proteins, amyloid proteins, involved in neurodegenerative diseases like Alzheimer's and Parkinson's. And this has led us to propose that they are a major avenue that serves for the spreading of these pathogenic proteins in the brain. And because these proteins cause cell death, if we could block the spreading of the proteins through the tunneling nanotubes in the brain, we could slow down or cure maybe these incurable diseases. Unfortunately, these connections are very fragile and we know very little about them because it's difficult to handle them, it's difficult to study them. And what we have done in my lab, we have set up a new way to image them by using high resolution electron microscopy. By doing that, we now know more and we have a target to impair their formation and maybe to cure diseases.
So, given that TNTs are really fragile structures, we moved towards the use of electron microscopy in cryo conditions, where structural information could be obtained by imaging the cells while keeping them as close as possible to their native state. So we set up a workflow that combined fluorescent imaging, which we used to identify the TNTs that were connecting the cells, and cryo-electron microscopy and tomography to see their structure in 2 and in 3 dimensions.
So when we apply these cryo-electron microscopy approaches on tunneling nanotubes connecting neuronal cells, what we found was that what was a single connection, which we observed by fluorescence microscopy, was in fact made up of multiple individual tunneling nanotubes, which we called iTNTs, which seemed to be intertwined around each other. And inside of the iTNTs, we saw really long, straight actin filaments, and squishing them were these vesicles that were using them as railroads to be transported between cells. We also observed these tiny little connections which seemed to be connecting TNTs. We discovered that NCAD adhesion molecules were colocalizing with these electron-dense structures, presumably to hold stability for the overall bundle. Between iTNTs, there were also tips of iTNTs which were either growing or retracting from either end. We presume that these could serve as guidance for the formation of future tunneling nanotubes.
At that point, we asked whether the iTNT could be observed in other cells of other species, not just in the mice. So we tested human neuronal cells and we confirmed that iTNT exists in other neuronal cells. To check if organelles like mitochondria could move through iTNT, we performed time-lapse imaging and observed that mitochondria could move between 2 cells via TNTs. Using cryo-electron microscopy, we confirmed that the mitochondria was actually inside of iTNTs.
To test if TNTs were open-ended, we couldn't use cryo-electron microscopy due to the thickness of the samples at the contact zones. So we use a focused ion beam scanning electron microscope, which permits to mill a thin layer of surface, 5-10 nanometers, by sputtering. Repetition of milling and imaging of the new surface creates a 3D reconstruction of the TNT's contact zone that we can then segment with amila to create the movies that you can see in the paper. With our results, we show the existence of open-ended TNT, which could correspond to the functional TNT.